Okay, awesome. Thanks, Andy, for that. Not quite too embarrassing. So I am going to talk about mutation because we've been working on it for almost a decade, but lots of other people have as well. And so there's lots of stuff going on in mutation. And the main reason is that sequencing has gotten cheap, right? And so we can go out and with little more than a trio, which is essentially the basic unit of all mutation studies, but we can now get it down to one parent and a kid with long reads. You can go and you can estimate the mutation rate of any organism, right? Because you're just asking how many mutations does the kid have that the parents don't have? And it's been done in lots and lots and lots of organisms. So what I want to talk to you about today are some of the most common patterns, especially in mammals. So I'm going to really focus on mammals, tree of life, and then ideas about the processes that are dominating. So I'm going to talk to you about two major patterns, major types of patterns in mutation rate variation. One is among species and with age, and you'll see that those are really two sides of the same coin, or my claim is they are, and then between the sexes. Okay, so first, patterns about mutation rate variation, that is how the mutation rate has evolved between species and with age. So if we plot the per generation mutation rate, so that's on the y-axis here. So how many mutations are there per generation per base? So first of all, it's really low, right? Like 1 in several hundred million or a billion bases. And 2, up with the average lifespan of the organism. Right? So here are some primates. Here are some worms. It scales with lifespan. That is the general pattern that dominates across lots and lots of groups, not just across these groups. This is a study from Lucy Bergeron. There's a bunch of different clades on here, but if you focus mostly on the mammals, you can see the orange dots, the age of sexual maturity or generation time, the average age at which these organisms have kids, is the mutations they leave to their offspring. So that's across species. Each dot here is essentially a trio from a different species. If you look within humans and you plot the age of the father at conception and the number of mutations that the father is leaving, you also see correlation with age, right? So older fathers up here at 40 are leaving something like 80 mutations, for instance. This is Icelandic data, by the way, trios from Iceland. And younger fathers at age 20 are leaving around 50 mutations. So there's this correlation across species, the average number of mutations, and there's correlation within species. It's not just humans. People have done this work in chimps. So again, terminal age of chimpanzees and number of mutations they've left. I'm going to show you a few studies from my group that basically repeat the same statistics, same answers, but I'll give you a flavor of what we've been doing empirically. So our first project was actually in owl monkeys. That's what an owl monkey looks like. And we have these slightly more complicated pedigrees. You'll see that F2 individuals become useful for some purposes, especially in checking that we're not making errors in inferring mutation rates. In general, the kind of methods are the same. The studies I'm going to show you. So we sequenced here 30 individuals, each to about 37x coverage with Illumina. And then 50% of the final mutation calls are inherited by F2. So that's actually one of the great things about F2s is like I get to check with transmission genetics that my mutations are real because only half of them should be passed on. So we look in owl monkeys and we see that the mutation rate is 0.82 times 10 to the minus 8th mutations per base per generation. And the average age of parents in our study was 6.6 years. We can compare that to kind of a typical human study where you get 1.2 mutations times 10 to the minus 8th per base per generation. But of course, their average age is 30 in these studies. And again, in the owl monkeys, the age of the father is correlated with the number of mutations per base that they leave. Owl monkeys, it's true. We've done this in rhesus macaques, more complicated pedigrees. This is basically what's available, but in like these kinds of pedigrees, we can save a little money because this dad gets used twice. So it's just one fewer individual that we have to sequence. And again, now the orange dots are each trio within these larger pedigrees. You can see that the macaque mutations go up with age. Now the Icelandic data are plotted from humans are plotted as little dots. You can see it's basically like the same rate of accumulation in the macaques and the humans, and they're both going up with age, right? So there's something similar going on here. I actually hid this before, but this human study was done originally because we also see more autistic and schizophrenic offspring of older fathers. So one of the ideas in the field is that, oh, that's driven by the fact that there's more mutations in older fathers. Macaques are a model organism for the study of autism-like, autism-related phenotypes. We see the higher mutation rate in older fathers in excess of autism-related phenotypes in older macaque fathers, in the children of older macaque fathers. We've done this with domestic cats. Again, slightly complicated pedigree because that's what was available. And if you just focus on the orange dots and the orange line, you can see again that the mutations go up with age. And of course, they're able to reproduce starting here at like 6 months or something. So it's true across all of these mammals. And to me, the most interesting thing about this pattern is it raises this question of, has the mutation rate actually evolved among species? Right, so there's a mutational process, or cellular process, that's driving these mutations. And when we look across species, we see that the phenotype that we're measuring is definitely different among all of these species. But of course, they are all also reproducing at different ages, right? So again, here's my macaques and my humans. Like these macaques, it's really just one line. This bar is the human puberty age. This bar is the macaque puberty age. I'm just extending it so that macaques are little baby humans. They're having kids at 5. Rate actually evolved, or are we meant? And I think you could think about lots of function valued traits that are like this. We would never imagine going out to the forest and measuring the height of different trees, species, and measuring some of our species at 6 years and some of our species at 30 years, and then saying like, oh yeah, there's this different heights have evolved. So something has evolved about the life history, that's for sure. But in general, it's actually quite hard to tell whether the mutation rate itself has evolved. Because I'm going to talk a little bit about it later, we can take the mutation rate as a whole and break it down into different types. There's mutations. And so we generally summarize those as the mutation spectrum. Here's the mutation spectrum in owl monkeys. Some of them are CPG mutations, which I'll explain later. Here's the spectrum in chimps. Here's the spectrum in humans. They're a little bit different, right? You can tell that they're different. But actually, these mutations also accumulate at different rates with age. And so, again, the phenotype is different. But my analogy with trees, if I extend it to inside the tree, right, this is what all the cells look like at 3 months, and this is what all the cells in the same tissue look like at 1 year. And so they're also quite different, even though, but we're still measuring them at different ages. So there's this broader question of what's actually evolving when we go out and we measure these traits among species, which is interesting, but I'm not going to talk about that anymore. What I really want to talk about today, although the first part's important to understand all of it, is the difference in mutation between the sexes. So, the first thing you should know is that we can tell which parent transmitted a de novo mutation. We can do it one of 2 ways. The easiest way to think about it is, again, genetics. We see a mutation here in the F1. It's heterozygous, so we don't automatically have information about which parent it's from. But then if we have an F2, we can see which haplotype it's co-transmitted on, because we can easily determine whether it comes from the mom or the dad. And so then we can say that here my little made-up mutation came from this red background, which I guess was the mom. We can also use linked reads if our reads include an informative SNP, so that the 2 parents differ in that SNP, and then I can say, oh, my de novo mutation is on the same background as this. I know which parent it came from. And in general, we can do that for, depending on the kind of experiment, 15% and 75% of all of our mutations. So if we take the same Icelandic data and we break it up into who passed on mutations, we see that there's this huge male bias in humans. So these are the number of phase mutations inherited from dad. and these are the number of phase mutations inherited from mom. So it's really different, right? The slopes of the lines are different, and the human line, the male line is above the female line. Okay, so that's data from other groups in Icelandic humans. Here's our data from macaques again. So the colors switch depending on whose study you're looking at, but males are almost always going to be on top. But macaques are here, quite male biased. These are the macaques from somebody else's study. Again, another study by Lucy Bergeron. It's the same result. In fact, their mutation rate estimate on average is like within 2% of our mutation rate estimate. So, there's a lot of tricks to the experiments, but you get the same results if you do them right. And you can see, again, like extremely male biased. These are our results in cats. Again, extremely male biased. I'm now going to show you an exception. So like we have been going across the tree of primates and broader mammals looking for patterns of mutation rate evolution. These are brand new results, but there's this weird thing where they're female biased, especially at older ages. And actually we've checked these mutations from these moms up here with Sanger sequencing, and they're all real. So there's something interesting going on with IIs. It's a lemuriform strepsirrhine primate, but we're not exactly sure what's going on there. Okay, so again, there are these patterns we see in essentially all mammals among species with the age of the parents and also between male and female parents. So really what I'm talking about is what causes these patterns. What are the kind of common hypotheses that have been in the field for a long time, and then like what do we think? Because the point of this is going to be that the common hypothesis is not correct. So the common hypothesis, I will call the germline replication hypothesis. And it essentially goes something like this. This part is real. This is mammalian germ cell development. So here's the female germ cell. There are about 22 mitoses. That is a big guess, by the way. Like nobody really knows, but about 22 before female embryos are even born, and then no more mitoses. And then there's a meiosis right before an egg is ready for conception. So essentially 22 or 23 cell divisions. On the male side, there are also 30 approximately, who knows, mitoses. Like all of the estimates are based on the size of the germ cell mass. So it's a guess. And then there's a pause. And then when they hit puberty, mitosis starts again. And so the idea is that because mitosis starts again, there's a lot more cell divisions. And so then, again, if we get to this male who's 40, his sperm have gone through lots and lots of cell divisions. So we'll have more mutations because cell division is this thing that is driving it. And if we go to this 20-year-old male, it hasn't gone through as many mitoses. So here the estimate is 190, whatever that, 650. And so fewer mutations. And believe it or not, JBS figured this. He actually figured out that there's male bias mutation by looking at disease phenotypes in offspring. And so here's what he said. If the difference between the sexes is due to mutation rather than crossing over, I don't know why he thought it was due to crossing over, or possibly, but many explanations could be suggested. The primordial oocytes are mostly, if not all, formed at birth. So he knew the female side is basically done with cell division at birth, whereas spermatogonia go on dividing throughout the sexual life of male. So if mutation is due to faulty copying of genes and nuclear division, we might expect it to be commoner in males than females. Right, so he out that hypothesis in 1947, which is actually kind of amazing. So I'm going to represent this hypothesis, germline replication, and the patterns it explains, like this. So it kind of jointly explains both the paternal age effect, this effect of the pattern of with age, and the male bias, right? Because both, it goes up with age because there's more and more cell divisions, and it's male bias because there's many more cell divisions on this part and on this part, basically at every age. Okay, interestingly, everyone really stops here with the Haldane paper, but he had something else to say. He had another hypothesis. And he says, again, it's weird English. By again, he means, on the other hand. So on the other hand, the chromosomes in human oocytes appear to pass most of their time in the Pachytene stage. If this is relatively invulnerable to radiation and other influences, the difference is explicable. So now his hypothesis is maybe there's just something fundamentally different about the way that males and females package and protect their gametes, right? He doesn't know which one it is. So he says on either of these hypotheses, we should expect higher mutability in the male to be a general property of human and perhaps other vertebrate genes. It's difficult to see how this can be proved or disproved for many years to come. So I'm going to call his second hypothesis the faulty male hypothesis, right? They're packaged differently, and there's just more errors in males because they're not as well protected or they're not as well corrected when damage does happen. Okay, so we have now these two hypotheses. There's others, but focus on these two, and I want to talk about how they differ in their predictions and what data we could use, or that has accumulated over the 75 plus years since Haldane wrote that paper, that we could use to distinguish among them. So I think the main difference is how we imagine mutations accumulating through time. So in the germline replication model, there's this kind of accumulation of mutations early in development. Actually, before the germline is set aside, it's extremely mutation prone. It's not protected in the way that germline is protected once you've set aside the germ cells. So there's this little bit of increase of mutations. There's a few mitoses in both sexes. Then everything is quiet till puberty. At puberty, males take off because of continued mitosis. Females just stay flat. In the faulty male model, again, there's this error-prone stage before germline sex differentiation. And then it's just that males accumulate at this higher rate than females, but both are going up with time. So again, coming back to this kind of representation of the two, the faulty male model also predicts a paternal age effect because it's going up with time. That's just the normal accumulation of damage to DNA sitting in an organism, and male bias because males are just crappier. Okay, well, that doesn't help settle anything. So what I'm going to do is to try to walk through 6 pieces of evidence that I think mostly help to differentiate these two hypotheses. I'm going to tell you they're almost all in favor of the faulty male hypothesis, but none of them is killer, right? Like, I think for each of them, you could do pleading where you're like, okay, well, I just have to add like one more detail to germline replication. And I think that's reasonable. And it's really like in the accumulation of all of them that seems to suggest faulty males is a better explanation. And then I'll talk about some of the implications of that. Okay, so data that are inconsistent with the germline replication model. So the first one, which is most obvious, is that the male-female difference in mutation exists at puberty, right? So like, if it's this accumulation of mitoses post-puberty that's supposed to be the deciding factor, why do they differ already right here, right? Like that's quite a big gap. And males supposedly just started re-undergoing mitosis. It's not just in humans, so like you can see it quite easily in this 3-year-old cats. Of course, we can't actually see how many mutations there are using this experimental design before they start having kids. So people are working on that, but it essentially requires that you start sequencing single gametes. So that doesn't make sense, right? Under the germline replication model, they should start out at essentially the same value, and then they should diverge, whereas under the faulty male model, there's been things accumulating ever since germline set aside, germline specification. I mean, again, you could say, oh, well, maybe there's like a weird bump in males that pushes them away from females right before puberty or at some point in the past. Then that's perfectly possible. But I'm going to claim that on the face, the difference at puberty is not consistent with germline replication. The second thing, not obviously predicted by germline replication, is that there is actually a maternal age effect. Like, it is weaker, and it's understandable why Haldane wouldn't have been able to detect it using disease phenotypes. But like in essentially all organisms, when we have enough data, we see that female mutation rates are also going up with age, right? So there's humans, there's cats. In everything that we have studied, when you have enough power, you see that female mutation rates go up with age. And again, if everything is dependent on cell division and mitosis, that line is flat. Whereas if it's just damage and females are better at correcting them than males, then you expect some damage. And again, we should definitely include environmental damage, accumulating damage in the germline replication model. It's silly not to include it. So you could add that to that model, but on the face, it's more consistent with the faulty male model than the germline replication model. Next kind of data. So I'm going to claim that there is a relatively constant rate of mutation accumulation. And what I mean by that is that in the germline replication model, there's this clock that's determining the rate of mutation. And that clock is determined by mitotic cell replication. So the question is, how long does it take a cell to undergo one full cycle of mitotic cell replication? And it turns out in humans, the answer is 16 days. Okay, so if mutations are determined by the mitotic cell cycle, then we should see that mutations accumulate essentially at the same rate as that mitotic clock. And so here I'm actually plotting, this line is the cell replication rate, and this line is mutation rate. And you can see they don't really line up. I think you could easily kind of explain that by saying every cell is undergoing cell replication, which is true. And so it shouldn't exactly match because you have different cells undergoing different numbers of replication before they become sperm. But they certainly don't match. The data that really don't fit mitotic cell replication hypothesis is the fact that the rate of accumulation is the same. That slope is basically the same in all of these different species, right? So again, this orange line is the best fit line from macaques, and this line is the line from humans. And in fact, we know that the spermatogenic cycle is a third shorter in macaques than humans, right? So again, if it's this mitotic cycle determining things, we expect that that slope should be steeper in the macaques because they're cycling through those mitoses at much faster rate. So the rate of accumulation of mutations is certainly much more consistent with the female hypothesis. All right, next pattern. I mentioned CpG mutations earlier. So a CpG is literally a C followed by a G with a phosphate in the middle. We say CpG to differentiate it from GC content, which doesn't require that it's a dinucleotide. And the special thing about CpGs is that in mammals and in some other species, the C in this pair will get methylated. And when it gets methylated, it is very easily deaminated, and so you get a mutation. So the C becomes a T. So when you see CpG sites, you get this hypermutation. It occurs about 10 times the rate of normal point mutations, because the deamination happens so rapidly. The most important thing is it has nothing to do with mitosis, right? It is completely separate from replication because this deamination is spontaneous. And so if we look at CpG mutations, it should help us determine whether something is, I'm going to use the word determined twice, determined by cell division and replication. So here, again, is the accumulation of all mutations in human males and human females, and the cell division rate there. And then here's the accumulation of CpG mutations. So CpG mutations are actually quite male biased, right? Like it's hard to tell. They're actually slightly different, slightly less male biased, but there is both a female effect in both. There's a larger male effect in both. And that is not at all predicted by the germline replication hypothesis. Just as an aside, and working on now, because I think this is more accessible to people, if you don't have a pedigree, it is still actually possible to measure male mutation bias by looking at divergence data on the X and the autosomes. And this idea was first proposed by Miyata et al. In 1987. And we've been doing that across multiple primates for which we don't have pedigree data. And we can measure this ratio of male to female mutations. And we can look at C to T transitions in CpG sites. So those are the ones that are most likely caused by this deamination, and C to T transitions at non-CpG sites. And you can see, first of all, they're all male biased and across all of these different species. And they're a little bit less male biased than these C to T transitions over here. So like there is something different about CPGs, but they're male biased. Right? Maybe this difference is the difference due to replication, which should have some effect. But their overall is always just this higher mutation rate in the males. Okay, so CPG mutations not really consistent with germline replication. We had the opportunity to look at mutation in a species with hibernation. So there's a colony of grizzly bears up at Washington State University. And they have pedigree. We don't have lots of ages. Basically, all the parents are the same age, or were the same age within a year of each other of all of the births. So we can't really look at that. The interesting thing about grizzly bears and hibernating species is that the testes grow and shrink through the year, right? So basically right before hibernation, that's over here, they're very small. This is the testis volume. And then they actually reach peak mass right before they come out of hibernation, because that's when they're going to go reproduce. But during hibernation, they're not producing any sperm, right? So the idea, and it's not totally sure, is that spermatogenesis is just shut off. Okay, so now we have an organism in which the males are completely shutting down, maybe, mostly completely shutting down spermatogenesis, at least spermiogenesis, like no sperm comes out during hibernation. You can ask me later how I know that. And so like that suggests that if mitosis again is the thing determining mutation in the males, that like we should see much lower male bias, because every year, all of the males in our study were about 12, 11 or 12 when they had kids. They've gone through 10 cycles of shutting down spermatogenesis. But in fact, 22 of the 26 mutations were transmitted by the male parent, and it's not any different from the proportion you see in humans. Okay, so in this hibernating species, in which there's something fundamentally different about how spermatogenesis is happening and the size of the testis, there's just the same male bias as there always was. So again, I'm going to claim that's mostly consistent just with the fact that the males are at fault, not what's exactly going on with mitosis. The last piece of evidence that I think is actually the most convincing, and I had nothing to do with collecting, is evidence from somatic mutations. So first of all, the somatic mutation rate is much higher than the germline mutation rate. Like there is something fundamentally protected about animal germlines. And if you look at mutation rates in the soma, they're an order of magnitude higher than in the germline. But if you look at somatic cells that have gone through different numbers of cell divisions in the same tissue type, because there's also a tissue type effect, what you see is that there's no difference between stem cells and differentiated cells, right? So they've gone through vastly different numbers of cell divisions to get where they are. And they have essentially a number of mutations that's determined by the age of the individual, not the cell divisions. Similarly, if we look at neurons, don't worry about the healthy versus Alzheimer's. There's no difference there either. Neurons do not divide. But age is still the thing that's determining how many mutations there are. Right? So this basically tells us mechanistically that we can separate the act of cell division from the act of accumulation of mutations. I think like fundamentally there should be an effect of cell division, right? Like polymerases aren't perfect, but it is undetectable by these studies. Okay, so here are all the pieces of evidence. I mean, I think, we started here for many years, and then genomics allowed us to like see all of these things. And so I want to talk a little bit about what the faulty male model says and what it means for our understanding of mutation accumulation, disease, evolution, all these things. Right, so here's Haldane's view of what I call faulty male. There is something different about male and female germlines. Either the females protect the germline better or repair germline errors at a faster rate. So, I mean, of course, there are lots of differences between males and females and the male and female germlines. There are lots of genes that are differentially expressed. One interesting thing is that they almost never compare germline cells in these studies because even though they're homologous, they're not really analogous. Like they're completely different. Of course, they express different genes, so nobody goes and measures how many genes they express differently. But overall, 37% of the genes in the human genome show sex-biased expression in at least one tissue. There are actually, I've looked, surprisingly few examples of differences that we know of that are sex-biased in germline function. This is basically the best example I could find. It's from worms. Polymerase theta works differently in the male germline and the female germline. It's a complicated story, kind of interesting, but there's a difference in how the molecules actually repair DNA and copy DNA in these organisms. I think the most striking difference, if you don't already know this, between male and female germline in many mammals, many animals, but not all, but all mammals, is that in the male germline, all the DNA, I'm out of batteries there, but at the top, all the DNA is wrapped around histones, and then in males, you unwrap it from the histones and you rewrap it around protamines to package it into sperm. And the female germline just stays wrapped around histones. So there's this one step. It can't explain maybe it explains the distinct between males and females. There's just one step where we completely repackage the DNA. And there's probably a lot of errors that happen during that step because it's all just sitting out there unwrapped. We also know that there are differences in how mutations accumulate with age and in specific types of mutations. So I showed you this figure up here before of the mutation spectrum. That's like, again, the summary of what's happening, but we can look at every one of the mutation types with age. And what you can see is that they're different, right? So the A to Gs kind of look like the overall picture, but the A to Ts, there's not as much difference, and the slopes are similar between males and females. So there is something different in the germlines of males and females that is causing these individual mutation types to accumulate at different rates through time. And I kind of alluded to it before, but there are huge hormonal differences between many males and females that start essentially at germline specification. And so you now have these circulating huge differences between the genes that are expressed because they're downstream of all these sex hormones. And remember, there's this both high mutation rate in both males and females, and they're the same before germline specification. So before those sex hormones are turned on, all of the mutation rates are the same, as far as we can tell, between males and females. So here's a little figure that Richard Wang in my lab made, which is trying to show like a summary of mammalian germline development. So there's this important step, pre-PGCS and post-PGCS is primordial germ cell specification. That's when you set aside your germ cells and the sex hormones kick on. And then you have the rest of embryogenesis, and then you have what happens basically post-puberty. And the faulty male model says, as soon as you now specify the sex of all of the cells, you get more errors in one than the other. And just for kind of perspective, the germline replication model is really saying everything's the same before puberty. And then after puberty is when the differences start to accumulate. And I think that is like actually different. Where the difference between male and females kick in. So I'm going to spend the last few minutes talking about what that implies, right? So if all of these things are true, if the faulty male model is true, that you have this difference in germline protection, in DNA error accumulation, starting basically after germ cell specification, what does that mean for lots of other phenotypes? And I think the most interesting implication is whether the male soma is faulty, right? Because in this model, we suddenly have a difference, like some underlying molecular difference between males and females when we have the specification of sex. Okay, so that's coincident with setting aside the germline. But now, that means all of our cells could be different between males and females. People didn't really think about, somatic differences between males and females because they were fixated on the germline replication, right? And if it's germline replication that determines the difference between males and females, then we don't really have to worry about what's going on in the soma because there's no difference in cell replication in the soma between males and females. But it turns out there are lots and lots of things that are consistent with faulty male soma. That is, the processes that are causing more mutations to accumulate in the male germline are also causing more mutations to accumulate in male somatic cells. So I think the most extreme is that there are huge differences in cancer rates between males and females. Now, many of these are due to gendered behaviors, right? Like the top ones are lung, skin, colon, liver, stomach. I mean, that's like smoking and drinking and eating meat. But it's not, so this is from one study. There's another study, which is again, male excess mortality is on this side of one, male excess incidence of cancer is on that side of one. All of these cancers are male biased. But I think there is something underlying that's not due to behavior. And so here's the rate of incidence of childhood cancers, and they're also male biased. Right. So there are gendered behaviors at young ages, but they don't involve dip and alcohol. And yet there's also this difference here. And so like many people have noticed the difference in cancer accumulation, and there are lots of ideas for why that is, and they all might be true. And I'm proposing that maybe also there's this accumulation of extra mutations in males, just nucleotide mutations. Besides cancer, stop somebody from calling me. Also, big differences in mammals in aging and senescence between males and females, right? So here are life expectancies, depending on what year you were born in, and like the female line is always higher than the male line. It is a general pattern in mammals that females live longer and have lower rates of senescence than males. This is a slightly confusing picture that shows basically the same thing. Now it's the death rate on the y-axis, but you can see that basically the line on the male side is always higher than the corresponding line, even by a little on the right side. So it's age stratified, right? Controlling for age, age-adjusted rates. I should have said the cancer was all age-adjusted rates also. All these age-adjusted rates are higher in males than females. And actually, so this is interesting. These are the top 15 causes of death. And the male to female ratio is higher in males for 14 of them. The only one it's higher in females for is Alzheimer, which has like a fascinating social policy, and is actually going away in more recent generations. But anyway, like there's this difference between males and females. And again, there have been lots of ideas published about why male mammals age. Mammals, there is some sorts of things that have to do with like large scale mutations, but not many that are like it's the point mutations that are causing the deterioration of these cells. At the same time, there actually is a somatic mutation theory of aging, which is not specifically applied to males and females, but is the general that we age, that senescence happens because of accumulating mutations in all of our tissues. Okay, so those are all like really indirect ways of asking, is the male soma faulty? What I really should do is go out and measure somatic mutation rates. It turns out that people have done that, but only incidentally. There are tons of somatic mutation rate studies. Most of them are not set up to compare males and females. Like the sampling is completely bad at that. However, if you look across all the cancers, and almost all of these are from cancer cells, but not the driver mutations, what you see is that men accumulate mutations in those cells at higher rates than women. Okay, so here's one set of studies. It's actually 3 different sequencing centers. So they're stratifying by sequencing center in case there's a difference in their ability to detect mutation. Here's another study that, again, is across different kinds of cancers. And so the important thing in each of these panels is that the blue average is a little bit higher than the red average. Both of these data sets, the mutation rate in males is not really as stark as germline difference, which is like 3 to 1, but it's always higher in all of these. But there's a lot of noise, right? Those are not big differences. So is the male soma really faulty? I would say we don't really know, right? There aren't well-designed experiments that get at this, even in humans where there's tons and tons of data. The most kind of fascinating data set I think that's out there is that there are some studies, especially Utah cohorts, where they know fitness, fecundity of individuals, and they know the mutation rate of those individuals. And there is a correlation between having higher mutation rate and having lower fecundity. So there can be this connection between mutation and fitness. And there can be this connection between germline mutation and essentially somatic fitness. Okay, so in conclusion, Haldane was a really, really smart person. He figured out basically all of this in 1947, but he couldn't really figure out how to distinguish between those 2 hypotheses. Now we have genomics. We can ask, right, interesting questions with those data. And I think that genomic data, especially from the germline, help us to essentially look at the empirical data and ask which of these hypotheses fit better. And I think there's more genomic data that we could collect. So, there are now comparative data sets on somatic mutation across organisms. And we can see that the somatic mutation rate is actually lowest in humans. So now what would be great is somatic mutation rate in males and females across all of these species and across non-mammals as well to figure out if this is a common set of patterns and processes. Okay, with that, I will thank everyone in my lab and at IU who helped with this work, the people at Baylor who helped with sequencing and analysis, the people who maintain the macaques and the cats and the bears, and thanks for listening.